Meiosis is the process which occurs in sexually reproducing organisms such as humans in order to be able to create sex cells. And this occurs in the sexual organs, so in the testes for males and in the ovaries for females. It's going to have some similarities to mitosis, but it's also going to have a few key differences. So I'll try and stress upon so that we can distinguish the difference. And the first key difference, which you might already be able to see in the way that I've set up, is we have two cycles of division. So we have prophase, metaphase, anaphase, and telophase 1, and then prophase, metaphase, anaphase, and telophase 2. So there's 2 rounds of division which occur, and this is what helps us to get from having diploid cells to haploid sex cells. Obviously, diploid being a full set of chromosomes, haploid being a half set. Our sex cells need to be a half set so that when they combine to create a zygote, we now have a diploid cell, a full set again, with half from the mother and half from the father. So what's going to occur in prophase 1 is we're going to start to see our centrioles moving to opposite poles. We're going to get some spindle fibers sort of starting to get ready to roll. And then our nuclear envelope is going to start to dissolve. Now, at this point in time, what we're going to have is some pairs of sister chromatids. So the DNA has replicated during interphase. We've got some pairs of sister chromatids swimming around in that nuclear envelope, which is starting to dissolve. Now in metaphase 1, here's one of our kind of first key differences between meiosis and mitosis. What we would usually see is all of those sister chromatids lining up in one big line along the metaphase plate. But in meiosis, we don't see that. They line up in their pairs, and then our spindle fibers are going to pull one complete sister chromatid to an opposite pole from each of those pairs. So along that metaphase plate, they line up in pairs rather than one big long line, which is what we might see in mitosis. So anaphase, we'll see something sort of like this occurring, where those sister chromatids, instead of being broken up straight away, they're going to be pulled to opposite poles in their pairs, like so. And then when we get to telophase, our centrioles have sort of receded off to the poles, something like that. We have a nuclear envelope reforming, and now rather than having 4 sister chromatids in each cell, we now have 2. So what we would see here is going from having 46 sister chromatids in this original cell to each of these two cells, when they divide in telophase and cytokinesis, will now only have 23 sister chromatids. 23 sister chromatids in each of those daughter cells produced through the first division. But remembering that these are sister chromatids, so they've still got their kind of double set of DNA. So what happens is each of these newly produced cells in meiosis 1, so cell 1 and cell 2, each of those cells will go through a second division of meiosis as well. So I've only drawn in one here just because you can imagine the process is going to occur twice. So let's say, for example, we're going to follow the path of cell 2 here. So cell 2 is now going to go through this process of division a second time, except this time it's going to slightly differ again, but it follows the same basic steps. So what happens is our first division will yield 2 cells, and each of those 2 cells will divide into 4 cells. So one full sequence of meiosis, we go from having 1 cell to having 4 haploid cells. So now in prophase 2, what's going to occur is we're going to start to see, I'll draw in these while I'm on red. Once that division has taken place and we've got a separate cell, nuclear envelope will once again start to dissolve, and we'll see our centrioles starting to move towards opposite poles and some spindle fibers starting to form, getting ready for our second round of division. So in metaphase 2, rather than our pairs lining up, this division we see looks a little bit more like meiosis, where we don't have pairs lining up, we've got them lined up along our metaphase plate, in one being long line as opposed to pairs. So now the spindle fibers will actually split the sister chromatids in this division, so that when we get to anaphase 2, we have our sister chromatids being split apart into now individual chromosomes. So in our final stage, telophase 2 and then cytokinesis, so once again our second division, we'll see our nuclear envelope reforming, centrioles to the opposite poles, and then this cell will divide to form 2. Alright, the first cell here will have divided to form another 2, so in total we end up with 4 haploid cells at the end of it. Okay, so the reason why these are the reason why we say these are haploid is because if we look at our original cell, this original cell had 46 sister chromatids, so it's kind of like 46 chromosomes but double copies. All right, so those 46 double copies then were able to become 23, I guess you could say 23 double copies. So we've still got the equivalent of, I guess, a full set of DNA. And then once they're broken apart, those 23 double copies, 23 sister chromatids, go to 23 single chromosomes, with 23 obviously being half of 46, meaning that we have a haploid cell. So there you have the process of meiosis, where a cell is able to be divided into haploid cells to form sex cells. And the reason why they need to be haploid is in order for them to be able to restore diploid number when a sperm and an egg cell fuse through fertilization to create a zygote.